Hello students, today we are going to discuss about two cell organelles and important one cell structure here. They are plastids, lysosomes and cell vacuoles. First of all, we will be discussing about plastids. Just like the previous class, mitochondria, as you know that it is a semi-autonomous organelle. Plastids are also called semi-autonomous organelle. Why semi-autonomous organelle? Because they are having their own DNA and ribosomes. Ernest Haeckel was the scientist who first discovered and named plastids. Plastids are present only in plant cell. They are absent in animal cells. Plastids are of two types mainly according to the pigments present in them. Leucoplast, these are colorless, no color is there. Chromoplast is colored. Leucoplast is mainly present in the roots of the plants and colorless areas of the plants. Leucoplast is again of three types. Amyloplast, which stores starch, aleuroplast, which stores proteins, and elaioplast, which stores fats and lipids. Chromoplasts are again of two types, green and non-green. Green chloroplast, green is known as the chloroplast, which is very familiar to you, which is responsible for the photosynthesis in plants, and non-green, they are carotenoid and anthocyanin. Carotenoid pigments are responsible for yellow and brown coloration. Anthocyanin pigments are responsible for blue and red coloration. Here is an ultrastructure of chloroplast, which is very important to discuss. Chloroplast is a double membrane structure. Here you can see this is the outer membrane, this is the inner membrane. Inside the chloroplast there are stroma, that is the matrix is present, and this type of stacks like structures are present, stacks of tubules are present, which are known as grana. So mainly chloroplast is made up of stroma and grana. And also you can see here circular DNAs are present, ribosomes are scattered here and there. And if one of the grana, that is granum, if we take it out, it looks like this. These stacks of structures, each stack is known as thylakoid. Inside the thylakoid, you can see that chlorophyll granules are present. And these are the stroma lamella by which the granas are connected with each other. In this way, chloroplast is formed. Plastids are known as kitchen of the cell. Specifically, chloroplast is known as kitchen of the cell because photosynthesis process is occurring here. Here you can see grana is the place where light reaction of photosynthesis occurs, and stroma is the place where dark reaction occurs. The colored plastid, that is chromoplast, they are responsible for coloration of different parts of the plant which are not green, like flowers, fruits, seeds, etc. They are helpful in pollination and dispersal of the seeds. That is about chloroplast. Now we are going to discuss about a single membrane structure that is called lysosome. Lysosome has got different names. Like very familiar name is, it is called suicidal bag due to its digestive function, intracellular digestive function. Christian de Duve is the scientist who first discovered this lysosomes in cell. Lysosomes are mostly present in animal cells. They are very rarely present in plant cells. Lysosome looks like this. They are very small. In ultrastructure, they look like this. They are actually formed in the RER. From here, the digestive enzymes are packed. Then they are going to the Golgi apparatus, from where they are pinched off and forms these lysosomes. They are characterized due to they are having almost 40 different types of hydrolytic digestive enzymes. They are mostly hydrolyses. Lysosomes are present in different phases in a cell. They are single membrane structure, as I told you earlier, mostly present in the animal cell. Due to their intracellular digestion, they can digest any types of foreign bodies which are entering inside the cell. This type of function is occurring in the white blood cells also. That's why lysosomes are known as digestive bags. They remove worn out cell organelles, cell debris. That's why they are also known as demolition squad or cellular housekeeper, because they keep the cell clean, and they are actually a waste disposal system which are present inside the cell. When a cell is damaged or during starvation, the cell is lost its function, then these lysosomes attach themselves and release the digestive enzymes, and then they digest the whole cell. That's why lysosomes are known as suicidal bag, and this process is known as autolysis or autophagy. They also help in the metamorphosis process, and when disappearance of the different structures in tadpoles like the tail and the another structure which is responsible for the aquatic adaptation, they are removed and gradually they form into frog. They also help in the process of fertilization when the sperm tries to enter inside the ovum to lyse the wall of the ovum. These lysosomes also help. Now we are going to discuss about the cell vacuole. Cell vacuoles are of three types mainly. The type of cell vacuole which are mostly present in plant cell and animal cell, they are called sap vacuole. They actually contain the cell sap. Here you can see the comparative position of the cell vacuole in plant cell and animal cell. In animal cell, the cell vacuoles are smaller in size. In plant cell, the cell vacuoles are attached with each other, forming such a big cell vacuole that they can push the nucleus aside and the other cell organelles also. Outside the cell sap, they are having a membrane, protoplasmic membrane, which is known as the tonoplast. Inside the vacuole, they are containing cell sap. Cell sap contains different materials, biomolecules, proteins, carbohydrates, nucleic acids, etc. They are also present here, and enzymes, hormones, and sometimes some waste materials are also present here. So these are called the sap vacuole. They also remain as the storage sac. They maintain the rigidity and turgidity of the cell. Here you can say in amoeba cell, protozoa cell or unicellular organisms, two more types of vacuoles are present, like food vacuole and contractile vacuole. Food vacuoles are formed inside the cell after entrapping and engulfing a food material inside it, and they help in digestion process. These vacuoles also help in unicellular animals for releasing the excretory materials. Here you can see that contractile vacuoles where the waste materials are collected, and after that they pinch off and they remove the excretory material outside. So in this way, these two vacuoles also help the unicellular organisms. So you can see here they also help in the osmoregulatory process and digestion of food and excretory process in unicellular organisms also. Here we come to the end of the discussion about plastids, lysosomes and vacuoles.